Hi guys, it's the Petri Dish Diaries here, and today we're gonna be talking about enzymes. Many chemical reactions can be speeded up by substances called catalysts. A catalyst alters the rate of a chemical reaction without being changed itself. Within any living organism, chemicals reaction takes place all the time. They are sometimes called metabolic reaction. Almost every metabolic reaction is controlled by catalysts called enzymes. Without enzymes, all the reaction would take place very slowly or not at all. Enzymes ensure that the rate of metabolic reactions are great enough to sustain life. A different enzyme is needed for each kind of food. For example, starch is digested to the sugar maltose by an enzyme called amylase. Protein is digested to amino acid by protease. Enzymes are named according to the reaction that they catalyze. For example, enzymes which catalyze the breakdown of carbohydrates are called carbohydrates. If they break down proteins, they are protease. If they break down fats, they are lipase. Sometimes they're given more specific names than this. For example, we have seen that carbohydrates that break down starch is called amylase. One that breaks down maltose is called maltase. One that breaks down sucrose is called sucrase. An enzyme works by allowing the molecule of the substance on which it is acting to fit into. The fit has to be perfect. The enzyme is like a lock into which another molecule fits like a key. We say that the shape of the enzyme and the shape of the substrates are complementary to one another. Product is formed and enzymes is released ready to bind to another substrate molecule. The induced fit model. In an enzyme controlled reaction, the substance which is present at the beginning of the reaction is called the substrate. The substrate binds to the active site of the enzyme, and the substance which is made by the reaction is called the product. An amylase molecule has a dent in it called the active site. This has a shape that is complementary to the shape of part of a starch molecule. On binding, the substrate and enzyme both undergo slight modification to fit better, because it has to be perfect. Here are the 6 properties of enzymes. All enzymes are proteins. This may seem rather odd because some enzymes actually digest proteins. Enzymes are biological catalysts. They are not changed in the chemical reaction which they control. They can be used over and over again, so a small amount of enzyme can change a lot of substrate into product. Enzymes are specific. This means that each kind of enzyme will only catalyze one kind of chemical reaction. Enzymes work best at their optimum temperature. Enzymes which are found in human bodies usually works best at about 37 degrees Celsius. Enzymes are made inactive by high temperature. This is because they are protein molecules, which are damaged by heat. The effect of temperature on enzymes. The increase in temperature increases the rate of the reaction. The increase in temperature increases the kinetic energy of the molecules. More molecules bump into each other. However, at very high temperature, above 40 degrees Celsius, for human enzymes, the enzyme gets damaged or inactive permanently, and they are called denatured. This is because the enzyme loses its shape and the substrate cannot fit in the active site. The effect of pH on enzymes. Usually, the change in pH affects the rate of the reaction. Most enzymes are in the correct shape at about 7 pH. At very high or low pH, the enzyme gets damaged or inactive permanently, and they are called denatured. This is because the enzyme loses its shape again, and it cannot fit. That is this for the video. Thank you guys so much for watching, and remember, biology is all around you. Petri Dish Diaries out.